Доброго дня, шановні колеги, всі присутні.
Good
Ви
afternoon, dear
підключаєтеся до вебінару
colleagues, dear
«Мікробіологічне
participants.
забезпечення біологічного матеріалу
You are
знань»,
present
який
at the
проводить Центр
webinar
громадського
dedicated to
здоров'я
microbiological
спільно
study
з
of
партнерами.
biological
Наразі
material
ми
from
чекаємо,
wounds.
доки слухачі
And while
доєднуються,
other people
і
are
буквально
joining
через
us,
хвилину ми будемо починати
we're
наш
waiting for
вебінар.
them and we're starting in a minute.
Ще раз вітаю, шановні колеги. Ви підключилися
Hello
до
again, dear
вебінару
colleagues.
Центру громадського
You
здоров'я, мікробіологічне
are
дослідження
listening to the
біологічного
webinar,
матеріалу знань.
Microbiological Study of the
Сьогоднішній
Wound
вебінар є
Biological Material.
продовженням циклу
Today's
вебінарів
webinar is the
Європейський
continuation
досвід
of the
впровадження
series of the
інфекційного контролю
webinars,
та
European
адміністрування
experience
антимікробних
of
препаратів у
infection
перспективі
control
для
and
України.
antimicrobial
Записи
stewardships
попередніх
prospects for
вебінарів ви
Ukraine.
можете
The recordings
знайти
of the
на
previous
YouTube-каналі
webinars are
Центру
available
громадського
on
здоров'я.
the UPHC
Цей
YouTube channel.
вебінар, ця лекція
This
буде
webinar,
записана і
this
також буде
lecture will be
викладена на
recorded
YouTube-каналі
and
Центру громадського
uploaded to
здоров'я.
the
Ми
YouTube
дуже
channel
вдячні
of the
нашим
UPHC.
міжнародним
We're
партнерам,
extremely
які
grateful to our
допомагають цій
international partners
організації
who are helping
цієї
us to
серії вебінарів.
organize this
Хотілося б
series
висловити
of webinars.
подяку
I would like to
Центру
express
контролю
my
профілактичних
gratitude
хвороб
to the
Сполучених Штатів Америки,
US
ICAP та
CDC,
Колумбійському
ICAP,
університету
Columbia
і
University,
організації
as
ЕСКМІД.
well as
І
XMEET
спеціалісти
organization,
цієї
which are
організації
the experts
долучаються до нас і надають професійні
involved
та корисні
in
лекційні матеріали.
our
Сьогоднішній
webinars,
спікер
providing
Агнес
very
Пál-Sonnevend,
useful
також представник
material.
організації
Today's speaker,
ЕСКМІД.
Agnes Pál-Sonnevend,
Отож,
also
сьогоднішня
represents
тема буде
ESCMID.
передбачено
Today's
висвітлення
subject
інформації щодо
will be
мікробіологічного
about
дослідження біологічного матеріалу
microbiological
зраду. Але перед тим, як ми
diagnosis of
почнемо лекцію, я
wound
хотів би
biomaterial.
скористатися з
But before we
можливістю і
start the lecture, I'd
проговорити,
like
що
to take an opportunity
наш цикл
and
вебінарів буде
mention that
продовжуватися.
our
Для
series
того,
of the
щоб
webinars will
долучитися до
continue.
подальших вебінарів,
And in order to
я вам
participate
рекомендую вам
in the future
підписатися
webinars,
на Фейсбук
I would
канал
recommend
Центру
you to
громадського здоров'я, де ми
subscribe
завжди
on the
завчасно
Facebook channel
публікуємо
of the
анонси
UPHC,
подібних
where we
заходів.
announce
І всі
the similar events.
наступні лекції також
And
будуть
all the
завчасно анонсовані.
upcoming lectures will
Ця
be
лекція
announced
буде
as well.
тривати годину.
This lecture will
І на
last
цій
for an
лекції
hour,
передбачений
and
синхронний переклад.
this
Тому ми
lecture
вам наполегливо
simultaneous
рекомендуємо
interpretation is
зосередитись
available.
слухати цей
So we
вебінар
recommend you, if you
англійською
want to
або
listen
українською,
to
включити
this
переклад.
webinar in
Інструкція щодо
Ukrainian,
включення
please use
перекладу
the
наведена на
interpretation
слайді.
option. And
Для цього вам
you can
потрібно
see on the slide the
натиснути
instruction of how
на
to
комп'ютері
use
планетку і
interpretation. For
обрати
that, you need
українську
to
мову. На
press
телефоні
the
може виглядати
globe-shaped
трохи
button
по-іншому.
and
Отож,
select Ukrainian
знову ж таки,
language or
попрактикуйтеся та
Ukrainian
включіть
channel. So
переклад,
please
і
try
ви
and do
можете
it.
слухати в
Switch on
оригіналі.
your interpretation, or you
Після
can
лекції,
listen
наскільки
to
буде
the lecture in
дозволяти
English.
час,
After the
у нас
lecture,
буде можливість
if
задати
we have
питання
enough
нашому
time, we will have
сьогоднішньому
a
спікеру.
chance to ask questions to
І ми
today's
будемо вже
speaker.
переходити до
And now we
сьогоднішньої
are
теми.
proceeding
Хотілося б
to
представити
today's
досвідченого
subject, and I'm
експерта,
happy to
професор Агнес
introduce a very
Пál-Sonnevend
experienced
з Пецького
expert, Professor
університету
Ágnes
Угорщини. Вона
Pál-Sonnevend
є
from
досвідченим
Pecs
спеціалістом
University in
з
Hungary. She is
мікробіології, і
an
вона вже не
expert
вперше
in
презентує
microbiology, and
лекції.
this is
Тому
not
знову ж
her
таки
first
хотілося
time
б
when she is given her
висловити
lectures.
велике слово
So therefore, I
вдячності
would
за
like to express my
неповторне
huge gratitude
систематичне долучення до
for
навчання та надання
her
такої корисної
involvement and
інформації. Отож,
participation
не
in
буду
our
далі
studies.
затримуватись.
With no
Сьогоднішня
further
тема
delay,
передбачає таку
to
важливі
today's
питання.
subject actually is very
Мікробіологічну
important.
діагностику
The
інфекцій шкіри та
microbiological
м'яких тканин
diagnostics
буде
of
передбачено
skin and soft tissues
багато практичних
infection
рекомендацій щодо коректного
includes a
забору
lot of
біологічного
practical
матеріалу зраду.
recommendations with
На жаль,
regard
в
to
контексті
correct
продовження
sampling.
повномасштабної війни
And
в
unfortunately, in
Україні
the
це
context
є
of the
важливе
full-scale
та
war in
критичне
Ukraine, this
питання,
is
тому
really
що це
important and
напряму
critical
впливає
issue
на
because this
якість та
directly
своєчасність
impacts
лікування
quality and
поранених
timeliness
військових.
of
Отож, сьогоднішній спікер, професор Агнес
treatment for
Пál-Sonnevend,
servicemen.
я
So
передаю
today's
вам
speaker,
слово.
Professor Ágnes
Всім гарного дня.
Pál-Sonnevend, over to you, and everybody have a nice day.
Thank you very much, Ágnes, for the kind introduction. And although the subject is have a sad relevance, it is also very important in peacetime. So I hope you would follow the advice what I try to give to you, which is based on international recommendations. So this skin and soft tissue infection topic, regarding to this, I have no disclosure whatsoever. So if you could please advance the slide and to the next one, please. What I would like to cover very briefly the clinical forms of skin and soft tissue infections, and then how to sample wounds and how to transport. So what's the logistic of the samples? And in the laboratory, what a microbiologist should do to correctly process the wound samples, do gram staining, microscopy, what media should be used, and how shall we evaluate cultures. So to the next slide, please. As you can see, there are plenty of forms of so-called skin and soft tissue infections, from simple superficial lesions from pustules and papules to nodules to folliculitis, cellulitis, or even necrotizing fasciitis. And when we are in the laboratory, we usually don't know which clinical form of SSTI our sample is coming from. So if we have anyone in the audience who is a practicing physician and having direct patient care and not a laboratory person, I would kindly ask them to always mention what kind of form of skin and soft tissue infection was sampled, which we have to process, because this helps us looking for the right pathogen. The next one, please. When we evaluate wound samples in the clinics, we always have to consider the patient's past medical history, for example, whether she or he has diabetes or atherosclerosis or some kind of circulatory issues, whether he is doing gardening or fishing, whether he's abuser of intravenous drugs, such as, I mean, behavior-wise, it's a mild expression for such thing, or whether the patient traveled or was bitten by an animal, another man, or an insect, whether he has done some exercise in fresh water or seawater, the wound is contaminated with soil or sand, and what is the anatomic site of the skin lesion. So based on this, a seasoned clinician could guess what could be the causative agent, but it also helps the microbiologist if all of these data are mentioned on the request form. Can we proceed to the next one, please? So based on that, we have almost the whole microbiology textbook of infectious agents which can cause skin and soft tissue infections. It could be a bacterium, a dozen of different bacterium may cause a skin and soft tissue infection, even more. Certain fungi would cause skin and soft tissue infection, from the superficial fungal skin infection to deep infection caused by Sporothrix schenki or other molds. Then viruses can cause skin and soft tissue infection, also parasites, cutaneous leishmaniasis, acanthamoeba infection, schistosoma in the tropics. These can be all related to lesions in the skin. But next one, please. This webinar is to focus on bacterial infections. So I will not mention any of the fungal disease or viral disease or God forbid, schistosoma and things like that. So let's go to the next one, please. In terms of types of skin and soft tissue infection, bacterial ones, we distinguish between superficial and deep. And if the injury penetrates deeper than the dermis, then we say this is a deep infection, and that makes our mind set to think of not only aerobic but anaerobic bacterial infections as well. And in both cases, we may face an abscess or just a wound, not continuity of the skin. But if it goes deeper than the dermis, then we have to think of the anaerobic infections as well. And here comes my first question to the audience. So if we can do the polling, which was on the next slide. I don't know, Sarah, whether you can start the polling. So please, please click on that one which you mostly see. I mean, whether you get in your laboratory or you send to the laboratory simple cotton swabs, or you use transport medium, or you use this plastic thing, flock swab in liquid transport medium, or send tissue specimens or pus or any other, and you can mark as many as you want. I give you like a few seconds to complete this. And I see the warning that I cannot vote. How does it proceed, Sarah? Can we stop it?
It looks like we have about 50, 55 responses out of 200.
Okay, so let's wait like 10 more seconds and then. Not everyone is active in such polling. So let's see, please, because then we are short of time. Okay, so I guess that what I see is the same order as was on my English slide. So I see some people have only simple cotton swabs without transport medium. And I see the others, but I cannot ask the audience what are the others, but probably you can write it in the chat, what is the other. But you equally get aseptically taken tissue and aspirates and all these properly transported swabs. But 34% also uses the simple cotton swab. So I got as a take-home message. So let's go further, close the polling and see what would be the ideal way of sampling of wounds. The most important thing that the sample should be taken always before giving the patient the antibiotic therapy. That's the ideal situation. A, you take the sample, and the B step is, the second step is to give the antibiotic, initiate the antibiotic therapy. If it cannot be withheld until the micro result is available. And the optimal specimen, in case of a wound, especially the deep wound sample, is an aseptically taken tissue specimen from the advancing margin of the wound. That may contain the most live microorganisms, most live bacteria, compared to pus or drained fluid. But it's also possible that if you have a closed off and not yet draining abscess, that you aspirate the wound and send the syringe, which is covered with a cap, as you see on the slide, in order to maintain the anaerobic milieu. If not enough pus is present and the wound is sampled by a swab, the preferable way to use the cotton swab, because the flocked, not the cotton swabs, the flocked one, because these which are shown on the picture are plastic hedgehog-like structures which can collect more cells, bacterial as well as human cells, than the simple cotton swab. So this is a more sticky and more effective way of sampling if the tissue or the pus cannot be sent to the laboratory. And obviously, you always have to keep your swab under either the liquid or under the jelly-like transport medium in order to maintain the anaerobic milieu in case of deep samples. So if the superficial sample is sampled, then the simple cotton swab could be okay. But still, we like the transport medium not to allow the swab to dry, because if the transportation takes somewhat longer, then drying microorganism may not survive the transport, so therefore we need the transport medium. So I hope you will have access to such float swabs with liquid transport medium. But if the jelly-like thing with the cotton swab is available, that's even better than the pure cotton swabs, which, as I've seen on the polling, a third of the audience is still using. So that's about the sampling, and then comes the next question, the next slide, about the logistics. In a hospital setting, and even if you have an outpatient clinic, ideally, the aspirates and tissues should reach the laboratory within 30 minutes. And this is preferred for the best recovery. I know anyone who works in a microbiology laboratory or does clinical patient care suffers because of the logistics. So unless you have this tubing system and transport system within the hospital, this 30 minutes, and if your laboratory is not located at the same premises as the patient care ward, then it's really difficult to fix this issue. But the 30 minutes is preferred for best recovery, just to have an ideal situation. And we never should refrigerate or neither incubate at 37 degrees the sample taken. So room temperature, especially if you have the transport system, room temperature would keep your wound swab or tissue sample in a condition that bacteria can be recovered from them. These commercial transport systems, which was on the previous slide, are safe to keep for a day. So if the commercial transport system is used, then you have to deliver it within a day because they are shown to be able to preserve the pathogen for up to 24 hours. So that's a relief for all those of us who suffer because of the logistical constraints. Then the next slide shows you those samples which shouldn't be accepted. For the left side is the simple cotton swab, which cannot be processed for anaerobic culture. And the other one is the mistake when the tissue sample goes into formaldehyde. And of course, the formaldehyde fixed sample should go to the pathology and not to us. And if it has already been placed into the formaldehyde, then forget the microbiological processing, because for culture, it is not possible to recover anything after the formaldehyde treatment. If we receive the right sample, and this is a tissue sample, next slide, please, then we have to be sure that it is processed in a way that the most microorganism bacterium can be recovered from it. And therefore, you have to smash the sample in a way, either by using sterilized plates and scalpel and just cutting it and homogenizing it to some extent, or you use the mortar and pestle, or on the next slide, you have a specific device in a tube where you can smear the tissue sample. Or if you are really well equipped, then you have the so-called stoma hair sample, next one please, in which you can place the sample in a bag, a sterile bag, and then it's like using pedals, smashing with physical force the soft tissues. You cannot use for any of these, the bone tissues cannot be destroyed in this way. So they are usually immersed in liquid culture because the bone is too sturdy for homogenizing the bone sample with that. So there, in that case, you can't do anything else but immerse the sample in liquid culture medium. So skin and soft tissue infections can be handled with this. And then comes the next question with the polling, whether you as a laboratory person or on the receiving end, the clinician, do you get gram staining results? So whether your laboratory you are using is doing gram staining on one samples, if they do, whether they do it always or only if you ask them. And if you are on the laboratory side, you can answer it for yourself. But here you can only choose one of your, the real setting of yours. And I'm relying on Sarah to see how many samples are collected and answers, and then you can close the polling when you feel that it's enough.
We'll give maybe 10, 15 more seconds.
Okay. Okay, so on occasion, half of the group receives or provides gram staining and its results, and almost 40% does it all the time, and the few people who responded did not. I understand this. This is a very good thing, because the last time I did this seminar, the polling was not showing the same result. Can we proceed to the next polling, please? If you do the gram staining, when the laboratory reports on the gram staining result almost immediately, so within an hour of receiving the sample, only next day with a preliminary culture result, and or only with the final results, or for those who do not do gram staining, or there may be a laboratory which processes the sample for gram staining, but they do not give a report on the gram staining. So that's going to be the last answer. And then I'm waiting for the answers. And if Sarah thinks that we have enough, then she can close the poll. How do we stand? Super. Oh, lovely. So probably those who do not perform gram staining, they do not report on the gram staining, but the remaining of the people gave an answer. They do some sort of reporting, and the majority does the reporting almost immediately. So let's see what is the advice. How shall we report the gram staining, and whether we shall do gram staining at all? So can we proceed to the next slide, please? And the recommendation is that if you have a skin and soft tissue, in different words, one sample, you should do a gram staining no matter, and record the relative numbers of white blood cells, polymononuclear cells, epithelial cells, and if bacteria or fungi are visible, then the morphology as well. And in any case, we have to, when we evaluate the culture results, we have to evaluate our culture based on the number of white blood cells, whether it was full with epithelial cells, so it's likely a superficial sample, or whether any gram-negative was visible and not cultivated, that's interesting, and so on. So we have to use the results of the gram staining for the evaluation of culture. And the next one, please. And those samples which were collected invasively, what I mean, it was collected during an operation in the OR, or in a sterile way, aseptic way, from sterile sites, the gram staining result is already a result from a laboratory, a microbiology laboratory, and the gram staining should be read and reported as quickly as possible. So it's a matter of urgency. And those of you who said that they report it within an hour, probably they meant the invasively collected specimen, and they really give high priority to the gram staining. So for selected specimens, we really have to do almost immediate reporting. On average, it can wait probably to see. It only helps the evaluation of culture next day during the first preliminary reporting. Now, what can we see on a gram staining? Can we proceed, please? We can observe the number of polymorphonuclear cells. Next one, please. That will be and the next one. So we have few polymorphonuclears and no bacterium, and that could mean that the patient already received antibiotics, or very little amount of bacterium is present, or our pathogen is not stained by the gram staining. I will have another figure on such a pathogen. Then the next one, gram-positive cocci in clusters, mostly meaning some sort of staphylococcus. The next one, gram-positive cocci in chains. This could be, let's say, from an erysipelas caused by group A streptococcus. Next one, please. And the next one, gram-negative bacterium with polymorphonuclear cells. The next one, all sorts of bacteria, several different morphology with white blood cells. That means that's a mixed infection. It could occur in, let's say, an abdominal wound, or if the bowel got hurt, or a soiled wound may show the same. And the next one is more interesting when you have round-ended gram-positive rods without polymorphonuclear cells. This could be a Clostridium perfringens, because the Clostridium perfringens toxin would destroy the white blood cells. So by the time the sample is taken, hopefully the patient is already in the OR for debridement, and the white blood cells were destroyed by the toxins of the perfringens. So let's go to the next one. This shows some sort of blunt-ended gram-positive rods with spores. Few of them showing these holes in the middle. This can be bacillus, can be anthrax or not, or streptobacillus moniliformis. The next one is showing actinomyces or nocardia. Both can be the case. And the last one is when the gram staining is not showing anything, but the Ziernielsen shows acid-fast rods. So you can think of mycobacteria atypical, or Mycobacterium tuberculosis skin infection. That's also a possibility in case of skin and soft tissue infection. So this is what the staining can tell you. And then let's proceed to the next one. Then comes the question that what media the laboratory should use for cultivation of deep samples especially. And then we use a battery of agars for recovering aerobic organisms, such as blood agar and chocolate agar. And usually we use an agar, either McConkey or Reusing methylene blue, for selecting of the gram negatives. We also do enrichment, and we also use anaerobic solid media. This can be of your choice, but this is always supplemented with vitamin K and blood. And if you want, you can use agars which are selective for gram-positive organisms, or if you have a high prevalence of MRSA, you can directly inoculate your wound sample on MRSA screening agar, just to be quick in detection of the MRSA which would influence the treatment, the choice of antibiotic. And then we incubate these agars and the enrichment broth at 35 to 37 degrees Celsius in 5% CO2, and of course the anaerobic solid medium in anaerobic environment, and the enrichment broth at 35 to 37 degrees Celsius, and next day we subculture from the enrichment broth. And you may also add anaerobic enrichment broth if you are particularly interested in anaerobic organisms. Then after incubation, we have the primary culture. And next slide, please. Where are we? That's it, I think. The gram staining results would give you an idea how to evaluate the primary cultures. If white blood cells were present and you recorded few epithelial cells or none at all, then the sample is considered to be a good quality sample. And then what we have to consider whether your cultivated bacterial colony morphology is suggestive of a pure or a mixed culture. And we also have to consider whether the organism cultured is a common pathogen, likely a rare pathogen, or a pathogen which could only be a commensal, a colonizer, or a commensal which on very rare occasion may be a pathogen. And then let's go to see what are these categories. So next slide, please. So the common pathogens in a wound sample are Staphylococcus aureus, Streptococcus pyogenes, Streptococcus agalactiae, and Pseudomonas aeruginosa. And if you found such on a culture, primary culture, then you have to do, colony morphology is suggestive of something like that. Then you have to perform identification and antibiotic susceptibility testing. But I usually tell to my residents that if you see a beta hemolytic colony, be suspicious of something real pathogen and then do identification. So if we went further, then I have a case to present. So if you could proceed, please, with the slides. In this case of a 25-year-old soldier who had a soiled combat wound on his leg and experienced sudden pain and swelling and exudate appearing soon after the onset of pain, so really quickly. And then very quickly his skin turned to bronze color, to blue-black color with blebs and bullae. And therefore he was taken to the OR and operated on, and the debridement was done, and the tissue sample was sent to the laboratory for culture. But before the culture, the lab did the gram staining, and what the gram staining showed, gram-positive rods and very few polymorphonuclear cells. There were few, but only small amounts, so 1+. And if we went to the next slide, or not slide, but the animation, the next day, growth was not observed on any other aerobic culture media, but on the fastidious anaerobic agar kept in anaerobic condition, large colonies with double hemolytic zones grew. And then that was our case, but the Malitov, we have here the Malitov, but you can use any other identification method to identify Clostridium perfringens. So a classical anaerobic organism, which was not listed in the previous common pathogens. And if we went further, then we can say that originally this patient was started on a broad spectrum carbapenem meropenem. And because of the recommendations say that for Clostridial infections, you can use penicillin, intravenous penicillin and clindamycin. Basically the antibiotic therapy was de-escalated from the meropenem because there was no growth of gram-negatives, no growth of Staph aureus, nothing but the Clostridium perfringens. And that was suggestive by the clinical picture as well as by the gram staining. But until you can be sure, you cannot say that the meropenem, and the meropenem is a correct choice, just too broad. So you can de-escalate therapy based on the no growth on the aerobic medium. And then let's see what other not so common but still pathogens are present. So if we went to the next slide, please. That's a long list which I don't want to read loud, but you see that there are plenty of organisms which are well-defined pathogens, but we do not encounter them frequently. So, such as Vibrio vulnificus, when you expect marine exposure, or Pasteurella species in animal bites, Arcanobacterium, or even Corynebacterium diphtheria, which can be toxin producer, and that has to be proven just to know that has a huge public health importance. So there are abundance of pathogens which are well-defined pathogens, but we do not see them so frequently as the first four which was listed on the previous slide. And then there is a special group on the next slide, which are although well-defined pathogens, but we have to take special care in case we see the suspicion of such pathogens, which is Bacillus anthracis, Burkholderia, Pseudomallei and Mallei, Francisella tularensis, and Yersinia pestis. And what we have to do, and sorry for the silly figure, please proceed to the next one. Basically, you drop them like hot potatoes. You seal the plate and send it off to the public health laboratory after the presumptive ID, because they are possible bioterrorism agent, and this is all of them are highly infectious to laboratory workers. So you not necessarily have a biosafety cabinet, and even if you had one, you're not supposed to probably, I can say plainly, fiddle around if you have the suspicion of Yersinia pestis, for example, but better to send it to the public health laboratory who has proper safety measures to handle such pathogen. The next category on the next slide are those which are pathogens, but could be also colonizers. And what the guideline says that only process these pathogens if in pure or predominant culture. So if the majority of the colony morphology on the plate shows similar things to either a gram-negative, which is Acinetobacter baumannii. Indeed, it causes skin and soft tissue infection. It became famous in the first Iraqi war, where the soldiers were wounded, and the Acinetobacter caused the wound infection, and it was a really difficult to treat type of multi-resistant Acinetobacter. The other organism here is Staphylococcus lugdunensis, and I would say that if you cultivated Staphylococcus lugdunensis, this is almost like Staphylococcus aureus. Staph lugdunensis, when you do the coagulase test, is negative. Its coagulase is negative, but it can cause as aggressive infection as Staph aureus and doesn't behave like the rest of the coagulase negative Staphylococci. Then we go further, and we have those organisms which are chiefly commensal and only rarely a pathogen, and this is a potential pathogen. And again, you process them only if in pure or predominant culture there are other coagulase negative Staphylococci or all Enterococcus, and mind your word, all enteric bacteria, E. coli, Klebsiella, Citrobacter, Serratia, you name it. These are all commensals, only potential pathogens. Other glucose non-fermenting gram-negative bacilli other than Acinetobacter or Pseudomonas. Stenotrophomonas also falls into the previous category, by the way. Anaerobic organisms which may cause infection, but not necessarily the causative agent of a wound infection, such as Fusobacterium or Bacteroides, or in case of deep-seated candidiasis, the Candida can be also causative agent, but most of the time it's a commensal. And then let's go even further to those which should not be processed unless it's a pure culture. So if you have nothing else on the plate, just some sort of coagulase negative staphylococcus, and then you do identification, or non-anthrax, non-bacillus, which is a not bacillus anthracis, green viridans group streptococcus, and any other miscellaneous anaerobes. Only if it's a pure culture, then you process it. And then I have an illustration of this, what is predominant culture and what to do for identification and susceptibility testing. And this is on the next slide. This was in my laboratory. We had an abdominal wound which showed 2+ polymorphonuclear cells. There was no epithelial cells, so nice and clean deep wound taken in the OR. And we've seen gram-positive cocci, which are marked by the arrow on the upper part of the figure, and some gram-negatives as well, which I tried to mark with the arrow on the lower part of the figure. And so far, a mixed thing with polymorphonuclear cells. And then the culture showed, and because we have the MARDI, we identified everything, but I tell you the point. So majority of the colonies were E. coli or Streptococcus aeruginosus. And we had a few colonies of Bacteroides fragilis, Fusobacterium nucleatum, and Peptostreptococcus anaerobius, plus a bit of Candida, few colonies of Candida. And you may wonder what to do with that various gram-positives, gram-negatives, anaerobes, and so on. And if we went to the next slide. So what is suggested in such case? Report on the predominant organisms, or the chief ones, E. coli and Strep aeruginosus, and say for the remaining species, mixed anaerobic bacteria resembling of intestinal microbiota, also including 1+ of very few, or you can say few colonies of Candida albicans. And do antibiotic susceptibility testing on E. coli and Strep aeruginosus only, and not the rest of the anaerobic organisms which are listed on the previous slide. And what to do in general when more than 2 potential pathogens are present. Then the saying goes that if it's an aseptically taken sample, the workup should be limited to no more than 3 potential pathogens, and also exclude the presence of obligate pathogens such as Staph aureus, Streptococcus pyogenes, and Pseudomonas aeruginosa. So you may even write on the report that we cultivated E. coli, we cultivated Strep aeruginosus and anaerobics, and Staph aureus, Streptococcus pyogenes, and Pseudomonas was not present. Now, what to do in terms of susceptibility testing of anaerobes? If it is requested, you can do, but most of the cultures which yield anaerobic organism is a warning sign that you have to administer, let's say, metronidazole or other anaerobic coverage to the patient. But there is little evidence which suggests that the susceptibility testing is predictive of clinical and bacteriological outcome. So therefore, if you have a mixture of anaerobic organisms, it's not necessary to do susceptibility testing for each. And usually it would take long time. And by the time you finished with all these, the patient either discharged or worsened to the extent that probably he is dead. So that's the technical advice. And to sum up all this with the next slide, or on the next slide. So if your gram staining shows bacteria, but the culture doesn't show any growth of organisms in the aerobic environment, then check the anaerobic growth. If there is no anaerobic growth, wait for a day. Then you have the enrichment broth, subculture, return back your plates to the CO2 incubator, or need think of whether you need some special media to recover the pathogen. We had sometimes Campylobacter, but that's mostly from blood culture, where clearly you see the gram staining, but the Campylobacter is unwilling to grow in anaerobic and also in 5% CO2, and then you have to do the microaerophilic cultivation. The next one is when you have a clear picture, single colony morphology, pure growth, you identify the organism and perform susceptibility testing. If it has a commensal potential pathogen, only process if it is from invasively collected samples or good quality wound based on the gram staining. And if the gram staining showed you gram-positive cocci in clusters, and you know that this sample was taken in the OR in a sterile way, really aseptic way, and then you get a coagulase negative staphylococcus, of course you process it, you do ID and susceptibility testing, but if your gram staining shows zillions of epithelial cells, very few polymorphonuclears, then you think that this is just a colonizer and not necessarily a pathogen, and you can stop at the level of identification or even just saying coagulase negative staphylococcus, and if further workup is needed, please call the microbiology lab, and then by the request of the physician, because he still thinks that this is a causative agent, the susceptibility testing and detailed identification can be performed. Then the next comes where this is really messy. You have multiple colony morphology on aerobic plates, only process if it is a good quality specimen based on the smell, so no epithelial, very few epithelial cells, and plenty of WBCs, and even some bacterial morphology is visible. And if it's an invasively collected specimen, process up to 3 predominant potential or common pathogen. And don't waste time on the rest. If it's a non-invasively collected specimen, up to 2 predominant potential or common pathogens. Really look for those which are high in numbers in terms of colony morphology. So in a way, you don't just process everything which is growing, but you select the important ones which are likely to influence the outcome of the patient. So to sum up all this, the take-home message for microbiologists, but probably it's also useful for those who are attending patients, that for a microbiologist, it is, next slide please, it is very important to maintain a good relationship with the clinicians who are sending you one samples in order to obtain the information on the location, on the deepness, whether it's a deep sample or not, how the sample was taken in the OR or in a really an aseptic way, whether the antibiotic treatment was started earlier than the sampling or not, and all sorts of information can be recorded on the request form. So if you have this conversation between the clinician and the microbiologist, then the sample processing and also the evaluation is easier. Then you always have to do gram staining no matter, and report immediately when it is invasively collected specimen for sterile body sites. And if it's in any case, you use the gram staining result for evaluation of the primary cultures, as was told you prior this final slide. And you evaluate your cultures and just do not report anything which is on the plate. From coagulase negative Staphylococcus epidermidis with a sample which was full with epithelial cells and so on. So think about whether it's real pathogen or not, or commensal, but still likely to be a pathogen because it was a OR sample and so on. And therefore, you really need the information from the clinician. The next slide shows you my references. And with the next one, I would like to thank you for your attention. And I'm happy to answer questions if there is any.
Дуже дякую, професора Pál-Sonnevend.
Thank you very much, Professor Pál-Sonnevend, for very interesting lecture. That was very Very interesting, very brilliant, I would say. Colleagues, please ask your questions using the chat box so that we could ask those questions to the professor. I see one of the questions already here, but let me express my gratitude once again for your really outstanding lecture, and let me very much agree and underline that your summary, your conclusions are very important. Communication with the managing, practicing physicians are really important. It really gives you the understanding of the picture, of the situation with the patient, how and when and from where the sample was collected. And it is also possible to communicate about other nuances with the doctor, if necessary. And what we observe as a very common mistake is that we really want to assess the quality of your sample, and not everything that grows should be treated with antibiotics, because there might be some commensals, there might be some contamination. And if you look into the sectoral standards in Ukraine, in case there is a contamination, you should repeat the sample. You should not treat everything that grows. You need to treat something which is the actual pathogen that brings to a systemic response. So let me now switch to questions. So the colleagues ask about information about semi-quantitative calculation of microbial burden, which you talked about, I guess, in one of your slides. Is that possible?
Well, you try to, I mean, it's always relative, this 2 plus, 3 plus, 1 plus, and so on. So usually it is a subjective thing. The A for the gram staining, if you have really white blood cells reaching to each other, then you would say it's a 4 plus white blood cell or polymorphonuclear cell content on the 1000-fold magnification. If you have fewer, you may say 3 plus. You see a fairly large number, like on the figure I showed to you in case number 2. This is 2 plus. And if you have occasionally one in a certain power field, then you say it's 1 plus. So this is the quantification of WBC. The quantification of various bacteria, I mean, anyone who had seen a mixed growth can judge whether you have 5 out of, I don't know, more than 200 colonies on a plate, which are beta hemolytic. That's easy to spot, and then you can process for the presence of Staph aureus. And in this case, the quantification doesn't really matter. Staph aureus is there, period, so you process it. If it's, as we said, commensal potential pathogen, but the majority of the colony morphology looks like an E. coli, then you process the E. coli. So this requires some hands-on experience, I would say, which one to process. So you cannot be always extremely exact, but with a bit of practice, 1-2 years, you would understand the relative abundance of colony morphologies, I should say. Does it answer your question?
Дякую за відповідь.
Well, thank you very much indeed, Professor. Let's check if colleagues have any additional questions with regards to that on the chat box. But let me switch to another question, because we have some. I would like to keep up with the time. So colleagues ask, what kind of media do you use for anaerobes?
Okay, what we use for anaerobes is called fastidious anaerobe agar. That's the name, but you can also use Schadler, or you can use Brucella agar and then incubate it in anaerobic condition. All contain vitamin K, hemin, and extra blood. Then we also use anaerobic enrichment, which is the fastidious anaerobe broth, but you can use any other type which is described in textbooks. In my experience, and also about the availability of agar, the FAA, so the fastidious anaerobe agar and the fastidious anaerobe broth works really nicely. And in case you want to isolate bacteroides from abdominal samples, then we use bacteroides bile-esculin agar, specifically looking for bacteroides and relatives. But that's a selective agar, not to be used alone.
Дякую за вашу відповідь.
Thank you very much for your answer. And the next question, what reference values do you use?
For what? What do you mean reference value?
Це все питання. Я думаю, ми можемо...
Sorry, that's the question. No more clarification. If the author of this question does not clarify, we can switch to the next question, I guess.
If someone wants to see the reference, I provided my reference in the slideshow. So the book I am using as a reference is the American Society of Microbiology manual of processing microbiological samples. So that's the reference.
Дякую, зрозумів. Тоді...
Thank you very much. Got it. Okay, let me repeat that in Ukrainian to our colleagues once again. Colleagues, you have references in the end of the slides deck, so you can use that for additional information. And colleagues also ask, what day do you give the final results back?
Okay, so our laboratory information system is linked to our hospital information system. So what we do, let's say on Monday we get the sample, we give the gram staining result on that very day. If it's urgent, urgently. If it's not, then just at the end of the day. So there is a preliminary result, then comes the cultivation, then comes a first day primary culture result, and because we have the MALDI-TOF, we do the identification immediately. So bacterial names go back on the second day, I mean 24 hours after the receiving the sample. For anaerobic primary culture, we check it after 48 hours unless Clostridium perfringens is suspected. So that's still a preliminary, and because we subculture from the aerobic broth on the after 24 hours, and then read the anaerobic a day later, and from the anaerobic enrichment broth we subculture after 48 hours of incubation and read it plus extra 48 hours. So it's usually 4 to 5 days to have the final, but in between we always report whatever was available from the reading. So basically, one sample, if we received it on Monday, has a prelim report on Monday with the gram staining, Tuesday with the aerobic primary, Wednesday, if there was some aerobic with AST, with the AST as well, plus on the anaerobic report, or if there is no anaerobic growth, then it says it's pending, and the final comes out by roughly by Friday. Yeah. So it's 96 hours for the final one, but we continuously communicate. And if there are specimens which require longer incubation time because we suspect of a slow-growing organism such as actinomyces, or the patient had a prior treatment with antibiotics, and then we do the enrichment, and then just keep the enrichment broth in the incubator for basically 3 weeks, and we weekly subculture from it, so that could be even longer. But what I want to to stress that in each step, we don't hold the result, the preliminary results for ourselves, but it gets to the clinician, so we can already have some basis of conversation, whether it was positive or negative. The preliminary report is given to them. So it's not kept in the laboratory, and then by the time the patient deceased, you release the final. This is not the case. I hope it's clear.
Дякую за таку розгорнуту відповідь. Ви відразу відповіли на наступне
Thank you very much. You have answered already to several other questions in the chat. And there is one more question, and we will finish today's session. So the next question is, if we want to have gram staining and culture, do we use one sample, or do we obtain two samples, one for culture and the other one for microscopy?
No, we do the gram staining from the same sample. So if you have a liquid sample, such as a pus or any drained fluid, or if you have a tissue which is homogenized, then you put with a sterile Pasteur pipette 1 drop to each plate plus the enrichment broth, aerobic, anaerobic, and another drop on the microscope slide. And then you dry it, fix it, and do the staining. So it should be the same specimen. Because then you can evaluate the same specimen, whether it contained epithelial cells, WBC, and so on. No separate tube. Same lab number, same everything.
Дякую за відповідь. І останнє питання,
Thank you very much for your answer. And the final question is the following. If there are no conditions for anaerobic culturing, can we use microscopy to suspect Clostridia?
Yes, you can. But not all Clostridia seen under the microscope would cause a clinical infection, and if there is a clinical suspicion, that should rule. The best, well, I shall say, easiest way to do anaerobic culture or set up something is to have those box system with bags which can remove the oxygen from the milieu, and that's the simplest. And then you just keep it the closed box in your incubator, which is otherwise used for cultivation of urines or something like that. So no CO2 incubator. But yes, of course you can. You can, but you cannot exclude other anaerobic organisms causing similar symptoms. Or if it's a mixed anaerobic infection, you will not be able to know if the others are fewer in numbers than the Clostridium perfringens. But the suspicion can be raised with the staining, yes.
Дуже дякую, професор Pál-Sonnevend, за
Thank you very much, Professor Pál-Sonnevend, for your lecture and for your answers. You have covered a big number of questions. So now we are going to conclude our today's lecture. Let me thank Professor Pál-Sonnevend once again, to thank our colleagues from ICAP, Columbia University, CDC, interpreters, XSmith, and all the participants, those who do very important job and provide care. Please subscribe to the Facebook page of the UPHC in order not to miss the announcements of such lectures, and the recording of this video will be uploaded to the UPHC YouTube channel in the near future. So everybody have a nice evening and goodbye.
Thank you very much. Thank you. Bye-bye.